So yeah, so there will be homework assignment for today. Okay, so I forgot how many questions. I made it last week. It was like 38 question multiple choice and on like a couple responses. So yeah, that's I think what the homework will be. So you guys, I mean, it's already assigned. I mean, if you guys, you probably, whoever that was here on Monday who gave me the email addresses, guys, I already assigned. I mean, I sent you guys logins and all, so that you guys can go in and then start working on it if you guys can. So that's what's gonna be the homework, guys. I mean, it's again, those questions are all AP questions, guys. Okay, so those are all college board questions, AP. I didn't get those from like Barron's or anywhere else, guys. It's all AP questions. So keep that in mind, guys. Okay, so and FRQs. I mean, dude, like if you guys go to school, a lot of teachers use. I mean, Jericho especially. You'll see a lot of questions on those when you guys go to school in the first like couple days. Okay, so a couple exams like you guys probably take. So, all right. So let's finish off from you guys. I think we were working lipids, right? So today, guys, we're gonna finish up the macromolecules. Okay, so we have lipids and protein, and I think that's it for you guys. And then a carbohydrates also. And then after that, we're gonna go into the enzymes, okay? So the enzyme in terms of the free energy and the enzyme reactions and inhibitions, okay, competitive, non-competitive inhibitions, and also allosteric inhibitions. And so I think that's gonna be it for today, okay? So for today's class, if time allows, I'll try to move into the next topic, which is cell organelles, okay? So that's what it is. All right, so lipids, I think we talked about a few things about this already, guys. Lipids are known to be, I mean, they kind of, lipids are a little different than the other ones. One thing to know about lipids, guys, okay, is these are the other stuff we talked about, the DNA, okay, this is the only thing we talked about actually in class. But lipids, guys, among other stuff like carbohydrates and proteins, this is actually, guys, it's a non-polymer. Okay, so it is a macromolecule. It's a large molecule. We understand. Okay, that's what we know. But it's not a polymer. So that's the differentiation, guys, with the other stuff. So and again, first of all, guys, if you guys are cold, guys, bring a jacket. Okay, so these guys are being a little smart. They brought a jacket, guys. Okay.
Last time it's freezing.
Huh?
Last time it's freezing.
Yeah.
I learned the lesson.
So I don't want you guys to get sick, guys. So bring like a jacket if you got. I mean, real men, never get cold, guys. Okay. So, I don't know, but and especially if you guys get cold, guys, I understand. But I gotta get the air going. Okay. So COVID and all, guys. Okay. So. All right. So this is a macromolecule, as we know. But the polymer. So polymer, basically, you have to understand. I mean, DNA was a polymer, because polymer means, guys, you have to create from monomers. Okay. So for deoxyribonucleic acid or RNA, for example, guys, I mean, it's chain up of a smaller molecule like this. Okay. It's a chain up of a small molecule. So they have to have a little monomers. Okay. So which is what? Nucleotides, right? So nucleotide is a monomer for DNA, okay, which they chain up phosphodiester bonds, right? So that clears the distinguish between monomer and polymer. But lipids, a lot of times there's a different class of lipids that we talked about. Sometimes but the class is fatty acid. We talked about triacylglycerol. So when they talk about the lipids, they a lot of times refer to this one, okay? So this is, sometimes they call it triglyceride, okay, which we talked about, I think, last week, where we have 3 carbon molecules, okay, which is glycerol, which is the alcohol, right? So these are OH groups here. Okay, so that's what we call the glycerol, and there's a fatty acid chain, right? So fatty acid chain is what, guys? It's an acid, as we know, right? So long chain of carbon like this, and there are 3 of them. Okay. So that's what typically a triacylglycerol is made up of. And fatty acid, as we know as well, guys, this is one of these guys is a fatty acid, okay? So you gotta understand, okay, why am I not calling this it's not a polymer? Because it's made from one single glycerol, 3 fatty acids, okay? There's no like monomer here, okay? There's no, okay, so there's no such as monomer like this, okay, as we see from DNA, okay? So it's a macromolecule, we understand, okay, but it's just a large molecule, okay? And then, as we discussed, guys, I think probably discussed last week a little bit, when you make a bond between an alcohol, this is an alcohol, right, with the OH group, hydroxyl group, okay, this is an acid. So when there's a bond, okay, when there's a dehydration process or condensation reaction, as we call it, guys, the water pops out, and what happens, guys, there's a bond now between this, like this, right? So this bond in bio, we call this ester linkage, okay? So you guys should know, guys. So ester linkage in bio refers to the bond, and not every time, but the bond between the glycerol and the fatty acid is called the ester linkage, guys. And then when all 3 of these guys, the same thing, as we know, this is what we call the triacyl glycerol or triglyceride. I mean, you've probably heard that term somewhere, okay? So, so that's basically, guys, as we know, what we call triacyl glycerol. And this long chain, guys, because this is a carbon chain here, okay? All carbons here. Typically, they have like 24 carbons on this fatty acid chain, okay, somewhere on there. So you cannot get like 100 or some stuff like that. It's around 24 carbon chains. So I think we kind of talked about last week, guys, in terms of how the carbon chain looks like. Sometimes you get what we call unsaturated fatty acid. It's unsaturated fat, unsaturated. Sometimes we get saturated fats, right? So saturated or unsaturated depends on the saturation of the hydrogens here. So what it means, I mean, if you guys don't have a background in chem, is when they say unsaturated means what, guys? I mean, yeah, if any one of these guys have what? A double bond between the carbons, okay? Then we call this what? Unsaturated hydrocarbons, okay? Or unsaturated fat. So if all the carbons, every single one of these fatty acid chain carbons, okay? If they're all single bonds, okay? All single bonds, we call this what, guys? A saturated fat, okay? Saturated fatty acid chain, right? So basically, guys, this has been a question for SAT and AP as well, guys. So AP, guys, we'll see. You guys are assigned with the homework questions, which directly is in line with the AP questions, by the way. So you'll kind of see what kind of questions you get. But so a lot of times when you get a saturated fat, okay, you guys know what that means now, is what? Then these are typically animal fats, okay? It's like butter, for example, guys, like that, you know? Like when you guys, I mean, again, some of you guys are not like meat eaters, by the way, but you guys eat like cooked meat, you know, the oil that you get, guys, a lot of times is what? Saturated fat, okay? So people say if you eat a lot of fatty foods, or if you guys like those like marblings, when you guys eat like those like steaks, that's what you're eating these what? Saturated fat. It should be very healthy for you guys, right? Unsaturated fat, guys, is so there is a double bond. Okay, so this is typically for a plant oil, for example, guys, like olive oil, probably you heard, guys, you know. So that's, you know, so this is typically, when there's an unsaturated fat, typically these are liquids, okay? So, you know, typically liquid, right? These are what? These are just like, you know, lard, right? Like butter. They're coming as solid. Okay, so I think for the AP, you might want to know why, okay? So why is this a solid and why this is a liquid, okay? So the reason for this, I think you really have to know it, guys, because of the next chapter as well, which is the membranes and all. So the reason for that, guys, why like saturated fat found as a solid while this guy's a liquid, it's due to the carbon chain, okay? This is a carbon chain, guys, of the fatty acids, okay? I'm gonna draw 3 of them. So if these are all single bonds, okay, if these 3s are all single bonds, okay, what happens, guys? They're nicely lined up, okay? All 3 of them, guys, nicely lined up like this, okay? So as you guys should know, right? So what will happen is, because they're nicely lined up like this, okay, they're closely packed with each other, okay? They're closely packed with each other, okay, which create what, guys? Stronger, in bio terminology, van der Waals interaction, okay? It's not the right term. So van der Waals, I mean, you probably see this term maybe in the reading, okay? But this is what, guys? This is something we call in chemistry LDF, London dispersion force. Okay, that's a better term, guys. And AP Bio, they do not use this term, okay? Or College Board in general, guys, they don't use the van der Waals force. But when you guys go take MCAT or when you guys go take a different exam, when you guys, I don't know, your school teacher maybe, like Olympiad, they use this term, van der Waals. Okay, so a lot of times it refers to what? Something like this, LDF, right? So the non-polar, like these are all what, like carbon chains, they're non-polar. What happens, guys, more closer they're packed, the strength of attraction is stronger. So a lot of you guys took chem last year, guys, by the way, which means what? They can have what? Stronger interaction. So, which means if they have a stronger interaction, what happens, guys? Yeah, dude, they're more likely what? Becomes a solid. Strong interaction makes what? Solid. So that's probably the reason why, you know, these animal fats with the saturated, you know, carbon chains like this, they're mostly found as what? Solids. But now, what if they're unsaturated? What happens then? So if there is a double bond, okay, like here. So if they're like, you know, like cis and you know, trans, you know that guy's like double bond here. So what happens, guys? What will happen is, if there's a double bond, what happens, guys? If they said this becomes a little kink, okay? So what happens is, because of double bond, guys, what happens? I mean, this bond angle is I mean, again, like your chem, guys, is sp3 hybridization, just so you know. Okay, between the carbons, which makes like the bond angle is 109.5 degree angle. But now, because of this, it's 120 degree angle. Okay, so they're not gonna be nicely lined up like these guys. Like it kind of moves away from them, so it kind of goes like this. Okay, so basically what happens is, I mean, it's not gonna be a straight line. It kind of goes away, which means what? The LDF, okay, interaction between these carbon chains gets weaker. Because they're weaker, what happens, guys? Yeah, there might not be a solid, right? They're more likely to become what? Liquids. Okay, that's the reason why. They said, oh yeah, by the way, you know, plain oils, they're what? They're supposedly healthier for you guys because what? It doesn't get built up in your body because what? They're not found as a solid, but more like what? It's a liquid. Okay. So some of the things you might want to, your teacher might bring up, guys. AP, you know, I haven't really seen a question like this on the AP. Like, okay, so compare saturated versus unsaturated fats. Okay, I haven't really heard that. So, but it's good to know. Okay, and since a lot of you guys have background in chem, you know, you probably know what I mean by this, guys. 120 degree angle for sp2, sp3. You know, some of the things you might kind of recall from what you learned last year. Okay. All right. So, and also we talked about a little bit, guys. When you look at this, right? So, you know, lipids, and we can talk about it. You know, we can talk about the carbohydrates also as well, guys, and we can kind of talk about that along with this. Yeah, I'll keep it for them actually. You know, but again, in terms of the formula for this, guys, there's a carbon, there's a hydrogen, there's an oxygen, right? Between these guys, there's a carbon, hydrogen, and oxygen, and if you, as you can see, guys, ratio perspective, right? I mean, like, dude, like, look at it, guys. There's like only like 6 oxygens here, guys. And there's a bunch of carbons, and what? Like a bunch of these carbons, right? And hydrogens here. So the proportion wise, okay. So there's like, if the carbon, for example, guys, for all this is like 75, okay, then yeah, this is maybe, I don't know, maybe around 120 or 130, whatever. And the oxygen is what, guys? Like 6, okay? So something like this, a ratio perspective, okay? I'm just giving an example, guys, right? For lipids, okay, they do have carbon, hydrogen, and oxygen, okay? They can have like other stuff as well, like they can have it also as well. But just from the ratio, okay, for you guys, with the AP perspective, there's a lot less oxygen compared to what? Carbon and hydrogen, okay? So that's one of the things to look for, okay? Because these get a little confusing because carbohydrate, like sugar, right? They got hydrogen, carbon, and oxygen as well, okay? But the ratio is more like, 1-to-1 ratio. So that's one of the ways to tell which one it is, okay? So is it carbohydrate? Is it, one of these guys, like lipids, right? So what's the best way to tell, guys? Is what? Look at the ratio of what? Carbon to the oxygen, okay? Typically, they're 1-to-1 ratio or very close to it for the carbohydrates. But for lipids, there's a lot more carbon compared to the oxygen, okay? Ratio perspective, okay? Because again, the long fatty acid chain, okay? They're typically, sometimes, not every time, 24 carbons per chain. Okay, do you see that, guys? Okay, so now, but like typically, guys, I mean, I think we talked about it a little bit last week, guys. When we, when you guys eat like fatty foods, right, like these things, right? This is too big of a molecule in order to enter your cells. Okay, like oh yeah, what if you guys eat something fatty foods? The purpose is what? Like yeah, that's energy for your body, right? Energy for your cells. Too big to enter your cell. Okay, so they have to break it down. Typically, it's broken down into what? Fatty acid and glycerol. Okay, they have to break these phosphodiester bonds, and then glycerol and fatty acid will be entering your cell separately. Okay. All right, so the other class of the lipid, as we talked about, is what? Phospholipids. Okay, this is very important, by the way, phospholipids. Okay. So phospholipid, guys, I think this is where we left off last week, right? So in terms of phospholipids, the only difference of the phospholipids compared to like what we just talked about is that one of the carbon chains, so this is a long carbon chain, guys, that stays as is, but one of them is what? You get a phosphate group, okay? So this is negatively charged, right? And also, on top of this, guys, okay, so, so first of all, guys, that's what we call phospholipids, okay? Phosphate, and lipid, okay? So, yeah, but you still have two long chains of these guys, but one of them is replaced as what? As a phosphate group. But it's not only this, you got I don't know how to draw this, a choline, okay, on top of that. That's not important, guys, I mean, but something with the nitrogen, with the positive charge here. So I'm not going to draw that, guys, but again, that's what the structural phospholipid is, okay, which has a minus charge and positive charges, okay? And now you have like hydrophobic tails, like hydrophobic carbon chains. So we talked about it with you guys last week, right? Or if I forget, I mean, to you guys, but so if you look at this side, okay, of the phospholipid, which I'm sure you guys know where I'm going here, okay? There's a plus side, there's a minus side. Okay, which makes them what, guys? I mean, okay, so with a plus and minus, what happens? They are attracting. They're very what? This side becomes very hydrophilic, right? Because, as we know, water molecule is very polar. Okay, so what does it mean, guys? As if you look at water molecule, in terms of structure of the water molecule, as we know, there's what? These are partial, very partial negative. Okay, very polar molecule, very partial positive. So this being a plus charge ion, these are ion, permanent plus and permanent minus, right? What do you think will happen? They're gonna attract the water molecule. Okay, do you understand, guys? They have a permanent charges, okay? So this side, yes, I mean, yeah, they're water friendly, okay? While this side is what? If you look at this side, the carbon chain on this side, they're what? This side is what? Hydrophobic. They don't like water. Because why? These are non-polar side, right? So that is one of the things that we talked about, guys, where if a single molecule has a what? Hydrophilic, hydrophobic side, what do we call this, guys? The terminology is amphipathic. Okay. This is a term that you might want to know, okay, for AP Bio. Okay, so, oh yeah, which of the following? Like you guys, like, I think you guys probably have, we talked about it. There's a question on this one for the homework for you guys. For AP Bio as well, guys, you gotta understand it's called the antipathic, which means what? Okay, so a molecule has what? Both hydrophilic and hydrophobic side. Okay, such as phospholipids. And phospholipid is what, guys? I mean, first thing first, guys, they draw the phospholipid like this. Dude, I'm sure, unless you really never took any bio at all, guys, I mean, you probably have seen this somewhere. Okay, the structure of the phospholipids. So this is a phosphate side. Okay, this is where it's hydrophilic side, and this is the tail, okay, which it refers to as the carbon chains. Okay, so this is what, guys? And typically, because of the nature of this, guys, you know, this is what, guys? Yeah, becomes what? Becomes the component of what? Component of the cell membrane. Okay, so you guys probably we talked about it already, guys. I'm not sure about how. So one of the things you have to know. So more to come on this one, guys. But you gotta know what this is, because probably next chapter or probably next week we'll discuss a lot of the cell membranes and organelles. And then the other stuff is glycolipids. Okay, I mean the glyco means guys sugar. So like you probably have like glucose molecule here. Okay, which is the same thing. This is a glucose molecule, by the way. We'll talk about it right after this. This is what hydrophilic, right? So glycolipids, guys. Okay, so it can be used as basically nerve cells. Okay, so glycolipids is not a big topic, guys. It's not one of the things they really talk a lot. But this can be a component of this, the cells in the nerve cells. Okay, cells around the nerve cells. So you don't have to go too crazy on this one. And the other one you have to talk about is steroids. This is important. Okay, steroids is something that you guys have to know for AP Bio. Steroid, just like is part of lipids. Okay, make sure I'm the same, guys. Okay. How do you tell the steroid, guys? A lot of times, steroid molecule, the shape is so like cholesterol, for example, that's a part of steroid, and also there's a steroid hormones, a lot of steroid hormones, guys. Okay, so if I'm not mistaken, guys, a lot of these like estrogen, like testosterone, those are like steroids. Okay. So one thing to know about the steroid, okay, as we all talked about, okay, AP Bio, they will never ask you to draw the structure. They will never come, oh yeah, draw like, okay, lipid for me. No, they never do that, okay? But when you see it, when they show the molecule, okay, you gotta, oh, that's a lipid. Oh, this is like what it is. That's something you have to know, okay? I think same thing with the steroid molecule. When you see it, oh, I know what that guy is, okay? What we should tell is what? They always have, usually, 4 rings, okay? So they got some of their stuff going on here, but a lot of times they have 4 carbon rings kind of meshed together like this, okay? So that's one of the things to look at, guys. Oh, if you see this, okay, that's what? That's gotta be steroid, okay? It's a tiny molecule, a lot tinier than the stuff we just talked about, because they don't have that long carbon chain over there. So it's a lot smaller, as you guys can tell, okay? But now this is also one of the what? Lipids, which is hydrophobic molecule. And now you will hear the term steroid a lot when we start to talk about, basically the cell communications, where there's a discussion on communication where, okay, so the hormones, right? So one cell wants to interact with the other cell. How do they do it, okay? So there's the different types of the hormones. Sometimes they call it peptide hormone or steroid hormones, okay? So steroid hormone being a hydrophilic molecule, they can just enter the cell, okay? So again, because of the cell membranes made up of, the phospholipids, these hydrophilic molecules cannot pass through easily, okay? Only hydrophobic, small, tiny hydrophobic molecules can pass through, okay? Such as steroids, okay? So that's one of the things, guys, about the steroid, like cholesterol or any other steroid hormones we'll discuss, okay? And now, like how selective, okay, impermeable the cell membrane is with the polar molecules. I mean, that's some of the stuff we discussed, but again, any ions, like Na plus or K plus, guys, by the way, these ions, guys, they will never be able to pass the membrane. So like these are tiny, right? Like these ions are very small, okay? They're very tiny, like these are single atoms, right? They lost electron, right? Because of their permanent charges, okay, they will never be able to enter the cell on its own, okay, like cell membrane by diffusion. This is a little, so you gotta be very careful with this, okay? So it depends on the teacher, okay? So I had a student, guys, who, I think I forgot, like what school was that? It's one of the private school, like I forgot where that was. It depends on the teacher, okay? AP Bio? I forgot what bio it was, but water molecule. If dude, just lay down. If this is a SAT2 class, okay, SAT2 back then, guys, SAT2 College Board, they say, oh yeah, water can just come in, okay? Like cell membrane, guys, you can come in, diffusion, okay, all good, right? AP Bio, no. They can't just come in. Water molecule being very polar molecule, they cannot come in into the cell membrane that easily, okay? They need a little channel, okay, something we call, guys, so what about these guys? Can they never come in? Well, they can come in, right? What do they need, guys? They need a little protein channel. They have to go through the protein channel, right? And that for the water molecule, it's called the aquaporins. So every cell in your body, right? Yeah, dude, there must be water coming in and out, right? So every cell in your body has what? A lot of these aquaporins, okay? That's That is the protein transfer, like like integral protein, transmembrane protein, which can what? Let the water in or out. Okay. So again, so they need to water molecule needs to go through aquaporins, okay, in order for them to come in. Because why? Why can't they just come in like the other small tiny molecules, as a cholesterol molecule or steroid molecules? Because why? They are very polar. Okay. So AP Bio, you have to know these terms, aquaporins. Absolutely no question, guys, okay? This is AP. Okay. And yeah, we'll talk about this a little more, guys, next week. But I mean, but this depends on your teacher, okay? So if your teacher is like, so they might not even talk about it, okay? And they might say water just can come in, okay, diffusion. So you gotta, when you take a test from school, you gotta follow your teacher's way, okay? So I think one of my students mentioned about aquaporins on her test. She got it wrong, because she's like, where'd you get this from, okay? And then she even showed a textbook excerpt. She's like, this is like, read this. Still wrong. So you gotta be careful, guys. Just stay for your school exams teacher, guys. Just follow her stuff, guys. She's the king in that class, okay? And yeah, that's only thing we have to talk about is the lipids, guys. Okay, very straightforward. So just keep that in mind. But we'll talk a lot about, guys, the phospholipids probably next week. Okay. All right, let's move on to guys. Carbohydrates for you guys. Okay. Also, guys, and we talked about a little bit for the DNA stuff, guys. Okay. Carbohydrates. So carbohydrate refers to a lot of times sugar, right? So the typical formula is, probably you see this, right? C6H6O6. So as we talked about, that's a molecule, that's glucose, right? It does. I mean, so this formula doesn't mean this is glucose. I mean, so keep in mind, okay? So it can be a glucose. So this can be, fructose, galactose, or glucose. It could be anything, right? And what do we call those guys? So fructose versus, fructose can have this formula, okay? And also, same thing, glucose, okay, can have this formula, okay? So what do we call these two? We call these what? Structural isomers, okay? They can have the same chemical formula, but they have what? Different structures, okay? That's what we call structural isomers, okay? So a lot of times, guys, my recommendation is you have to know the shape of glucose. Okay, my recommendation is at least glucose. Okay, you have to know their shape. When you see it, you have to know what this is. Fructose, like whatever. Okay. So for AP Bio, guys, you just have to know the shape of a ring structure glucose molecule. That's all you have to know. Okay. So when you draw the glucose molecule, okay, so you don't have to draw it, it's fine, okay? This is, there's oxygen here, okay? Carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, and carbon 6, okay? So there is a number. This is oxygen here, okay? Carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6, okay? That's how you number a glucose molecule, right? So again, like typically, right? Like carbon, ratio perspective. Okay, carbon to hydrogen to oxygen ratio. It's 1 to 1 ratio, typically. Okay. And then, so what do you guys know about what do we use? I mean, lipids, right? I forgot to tell you. Lipids are what, guys? Lipids were for a long-term energy storage, okay? Like, if you guys eat a lot of food, if people eat a lot of food, what happens, guys? Yeah, they get big, right? They gain weight, right? Just like myself, during COVID times, you don't move a lot, right? You just stay from home, you work from home. So typically, guys, you're not that active, you eat a lot, you eat as before, what happens, guys? People gain weight, right? So how do they gain weight? So it's what? They turn the energy into lipids, okay? Our body. So lipid is for a long-term energy storage sometimes, okay? So I'm not gonna go too much into, guys, LDLs or lipoproteins. I'm not gonna do that right now. But lipids are for a long-term storage. Are these fat cells, adipose like tissues, okay? So their job is only one thing, which is to store fats. Okay, that's like you probably have seen like liposuction, for example. Like they take the like fat out. It's like you see all those little circles. I mean, those are fat cells. Okay, which contains lipids only. Okay, so and also the fat also gives you insulations and protections and all that stuff, guys. As you know, but what about this guy? This guy, carbohydrates. So these are what? For a short-term energy. Okay. Short-term energy, right? So if you guys go buy those like energy drinks, for example, guys, what do they have? A lot of what? Carbohydrates, right? So because why? Like short-term boost. So and like for example, guys, if you have a test, for example, okay, oh my God, I'm so tired. Yeah, like eat something with sugar, then it will kind of boost you up a little bit for like about an hour or two. Okay, because what? They provide what? A short-term energy for you guys. Okay. I mean, don't try to do that a lot, but. I mean, something like that, right? If you guys have an important test, you got like 2 hours of sleep, then yeah, like maybe some of these guys, sugary stuff might help you a little bit, okay? Like as some of the kids, right? Like if you guys were a little kid, or if you have a little younger brothers or stuff like that, and if you feed them something very sugary, what happens? They start jumping around, right? Like just because why? It provides them energy, short term, okay? So a lot of times, okay, when someone eats food, okay, the energy gets to the cells by what? As a glucose molecule. Okay? So this is how the cells are getting the resources, okay? So if you put a lot of these guys in there, okay, like in your diet, what happens, guys? Yeah, a lot of times it will just go straight into the cells and what? Give them energy. Okay? And that so understanding from that, right? So I think one of the stuff that you guys should understand is what? Where's the energy at? Okay, so in the molecule, where's the energy at? So a lot of people say, okay, it's in the bond. So I don't disagree, but you have to understand exactly where they're coming from. That's a chapter, I think in a couple weeks. Okay, so the energy comes from the carbon. Okay, as we discussed, right? Carbon is where the energy is being stored. Okay, so the electron in the carbon in these molecules are where the energy is at. So one thing that you should know, okay, or we talked about, lipids versus carbohydrates, right? If you have 1 gram of lipids, okay, and 1 gram of carbohydrate, sugar, okay, which one will give you more energy? What do you think, guys? I mean, you probably heard this before, right? Like, if you have 1 gram of that guy, 1 gram of this guy, what happens, guys? So, per mass, okay, again, lipids got carbon, carbohydrates got carbon, of course, like carbohydrate, right? But now they have carbon, hydrogen, and the oxygen. Okay, and this one has the same thing, same components. Okay, same components, right? Like they're made of the same thing. Okay, same elements, right? We talked about it, right? Where the energy is at is what? In the carbon. So okay, so if you look at it, the ratio is what? 1 to 1 ratio, right? What about for these guys? Like it was like what was the ratio here? It's like what, like 50 to like 100 to 1, right? That's a ratio, right? Because which means what? There's a lot more carbon, okay? Even if what? You have the same mass of the lipid and carbohydrate, right? Which means what? Lipids can give you more energy per mass, per gram, compared to what? Carbohydrates, even for a lot of the other stuff, such as like protein or stuff like that. So 1 gram of lipid can get you more energy compared to carbohydrates, okay? So long-term storage. Because why? Why? There's more carbon in 1 gram of lipid versus 1 gram of carbohydrates, okay? So one other thing to know, guys, with the carbohydrate stuff, guys, you have to know, like bare minimum, okay, for you guys. You guys have to know alpha glucose, okay, and beta glucose. You gotta know the difference, okay? These are what we call anomers. It's good to know. Your teacher will not mention this, okay? Anomers. You're probably not gonna say it. Okay. So the difference is, I'm gonna kind of have to redraw this, by the way, okay? So alpha glucose, okay? So let's talk about this one first. I'll be quick, guys. Okay, with this guy. So there's a first carbon, second carbon, third carbon, fourth carbon, fifth carbon, okay? This is fifth carbon, guys, the CH2OH, okay? It's a ring structure, okay? So if you look at this, So there is hydrogen with the OH, okay, with the OH here, hydrogen, OH here, hydrogen, OH here, okay. So if you look at this, okay, this structure right here. So this is a first carbon, okay, and this is a second carbon, this is a third carbon, this is a fourth carbon, this is a fifth carbon, this is a sixth carbon, okay. Whether you got an alpha glucose or beta, okay, is depending upon what? This one, C1, right there. You gotta watch for this guy, C1. So when you look at this, okay, when you look at this, guys, you gotta be careful. So my recommendation for AP Bio students, guys, is look at what C4 is and what C1 is. Okay, that's typically what you have to know. Okay, if there's a OH group here, hydroxyl groups. Okay, if they are on the same side, okay, you see they're at the bottom, guys. That's what. This is an alpha. This is alpha glucose. You understand, guys? And now, a beta glucose is what, guys? Beta glucose is basically what? Okay, so there's an OH here. There's hydrogen there. Okay, that's one thing to note. Now that's what we call, guys, a beta. Okay, keep that in mind. Okay. So, if I did mention, guys, beta glucose is more stable form. Okay, compared, like when you compare the two, beta is more stable. There's more beta compared to the alpha. Okay, so you know. So when the plants, they create these glucose molecules, okay, there's more of beta because why? This is more stable form. Why? Like, okay, so we don't have to talk about why. But one thing to know, like, what the hell, why? Okay, why there's more of this guy, okay? So, I mean, one of the things to note is what? Like OH groups here. They're what? These are hydrophilic, right? Hydrophilic, okay? Which means like they attract electrons and they repel the same charges, okay? So when you look at this one, okay, compared to this guy, okay, they're what? I mean, one of the things is what? They're kind of far away from each other. That makes them a little more stable. Okay, you can take a look at it in that regard for you guys, just so you know. But keep that in mind, okay? It's the difference between these two. You know, they will never say, oh yeah, why do we have to, like, they will never say that, okay? So if we didn't talk about it, guys, just so you know. So carbohydrates, guys, can come in as like disaccharides or, you know, polysaccharides as well, guys. They can chain up because these are monomers, right? They can chain up, you know. So if there is a second, okay, glucose molecule. So there's hydrogen here, there's a OH group here, there's hydrogen, OH group, this is alpha, okay? So what these can what? They can go into dehydration and what? They can bond and make what? What we call glycosidic linkage, okay? So this is 1,4, what? Like one, this is the first carbon, fourth carbon, right? 1,4 glycosidic linkage. Okay, and this is what? This is alpha glucose. You gotta say, oh yeah, alpha 1,4, carbon 1 and carbon 4, glycoside linkage. Okay, that's one of the things you have to know. So if many of these guys chain up, so if you have a 2 glucose molecule, what happens, guys? This is what we call maltose. Okay, maltose, right? So you gotta know at least like 3 of these disaccharides. Okay, like 2 molecules chained up. Okay, maltose, lactose, and also sucrose. Okay. So I think we talked about it. So if you look at, there's a long tail of these guys. What happens, guys? Turns into, like, plant cell. We call this what? Like, there's more of these guys chained up. This is what we call starch. Starch can be amylose or amylopectins. Again, I don't think that's needed for AP. Okay. It depends how they chain up. Okay. The mechanism is the same. Okay. They got all of them have an alpha glycosidic linkage. Okay. You gotta know that. So now, beta. Okay, if you guys have beta glucose. So beta is what, guys? Beta is where you guys have, I mean, if there is, okay, OH group is here, hydrogen is there. Okay, that's what we call beta. So that's the only difference, guys, by the way. But now the problem is, okay, with these two. So when they chain up, right, like when they're trying to make a long polymer out of this, the problem becomes like, as you can see, there is what? OH group is here for the first carbon, and what? This is hydrogen there. This, guys, need to line up, okay? in order to go into what? Dehydration. They can't because what? Dude, they're far away from each other. You understand, guys? They can't. So what do you do, guys? What do you have to do? You gotta flip one of these guys like this. It's the OH group is on the bottom. Okay? So, which means, if you look at it, so the oxygen is in the bottom here now, and what? The OH group comes along with it. So, in a way to show this, guys, right? So, it was like this, but now I gotta flip this guy like this. You understand, guys? That's what's happening. So every other one is what? Normal, flipped, normal, flipped. You understand, guys? That's what we call a beta glycosyl linkage. So this is what? Now these two guys can, water goes out, H2O goes out, and what? Create a beta 1,4, okay, glycosyl linkage, okay? And if there's a long chain of this guy, what happens? What do they turn into, guys? It just turns into cellulose. There's like cellulose, okay? Cellulose is what, guys? It makes up cell wall for the plants, okay? And we talked about it, guys. We do not have the enzyme to break this down, okay? So it's only for what? Like very typical stuff like fungi, some of the fungi, and also some of the bacterial cells can like digest cell walls like cellulose. We can't. That's why your parents put, eat your vegetables, because why? We can't digest it, it comes right out and it flushes your system, I guess, okay? Something like that, right? So one thing to note, guys, cellulose, and that's a beta linkage. You have to know this with AP Bio, okay? They will ask this question, AP Bio, your teacher will, okay? And I think that's more you guys have to know. And I think some of the other folks, guys, we talked about, there's a term like, L, like D, stuff like that, like, dextrose, like, that is not for AP, just, Your teacher can go, your teacher probably will not go above this point, for the carbohydrate perspective. And short-term energy, and polymers can be, like what we talked about. It could be cellulose, it could be a long chain, as we know, okay? All right, so the most important part of this chapter, okay, is basically protein. That's the most important thing. That's what the AP will ask you, okay, 100% of the time. So these, maybe a multiple choice question here and there, but in mind, guys, a lot of times the chances-wise, the questions will be based on protein. So let's talk about it. So protein, first thing first, guys, let's talk about the function of protein, okay? Protein can be a structural protein, okay? Or sometimes they call it globular protein, okay? Globular or structural. Like structural protein can be like keratin, protein in your hair, which only stands for what? Structure only, okay? So it gives you the structure, rather than other functions, right? But now globular is what, guys? Like as you can see, globular is what, guys? This is the enzyme where it's a little glob and what? A lot of times functions as an enzyme. Okay. Enzyme reactions, okay? So why is this important, guys? Act on enzyme, because again, in bio, right? So it's a little different in bio, guys, okay? They want to have a lot of times, guys, for the reactions that occurs in the cell, guys, they want to have control. Which means that they want to stop a reaction when they can, they want to let the reaction go when they can as well, okay? So to provide that control, there's an enzyme, okay? So, which means, guys, in bio, a lot of times, if a reaction must carry on, if the reaction must occur, there must be what? Enzyme present and activated so the reaction can be what? Can be pushed further, okay? We'll talk about it, guys, right after this, the protein stuff, okay? Like the free energy and all that stuff, okay? So, keep in mind, guys, dude, think about it, there are what? Thousands and thousands of reactions that happens in a cell, in your body, okay? There's many of these things, okay? And there is what? One enzyme should be controlling one reaction, typically, right? So, which means what? There must be, thousands and thousands of what? Different enzymes available in the cell, okay? There are a lot of the enzymes, they're very different. So, how do they know, okay, so how do enzymes know, okay, so how do they know my role is activating this reaction. How do they do that? That's the question you have to ask yourself, okay? And proteins, right? So a lot of times their role is, okay, not for typically for energy per se, like lipids and carbohydrate, they're what? Providing energy for yourself, okay? So energy storage or short-term energy usage. Protein, no. They are what, guys? They're not there for you to use energy, okay, out of them. But like, I'm sure you've seen some of the folks, guys, unless like you're not eating at all, okay? Unless you're not eating like your food, or people like poorer countries, the body is using what? Like, yeah, protein to get energy, okay? But a lot of times protein is the last source of energy, so okay? They don't use it. So one of the reasons is if the protein gets processed, okay, they got like, what do you guys protein? They have nitrogens in there, okay? And nitrogen, right? So when nitrogen gets processed, what happens, guys? What happens? It produces what? Nitrogenous waste, okay? So when there's nitrogen, what happens? I mean, dude, like we produce what? Our body produces urea. So if there is a lot of urea build up in your body, what happens, guys? The pH goes up. Because again, nitrogen is what, guys? It raises the pH, okay? So which means, yeah, your body, like, yeah, like they don't want it, okay? They don't want to produce that much nitrogen waste, okay? So protein is not, it's not the ideal, okay, resource for energy perspective, like not for like daily energy usage, okay? So protein, right? So enzymes, right? Like that's the ultimate thing. Okay, so well, how do they produce this specific enzyme, right? So you gotta understand a few things, okay? Which is proteins have structures, and this is very important, okay? Primary structure of the protein, okay? Secondary structure, and tertiary structure, and quaternary, okay? Structure of the protein. So you gotta know all four, okay? These structures of the proteins. You gotta know all of them. So protein is made up of what we call the monomers of amino acids. I'm sure you guys all know this, right? Amino acids. So the term amino acid, guys, comes from amine and carboxylic acid. Okay, this is the structure of the amino acid. Okay, there's an amine group here. But this is an amine group. Okay, the functional groups, guys, you get to know. Amine group here. This is carboxyl. Okay, carboxylic acid. So why is it called amino acid? Because why, dude? Like amine, acid. Okay, right there, acid, right? So this is called amino acid because why? Yeah, dude, all of these, amino acids got what, guys? Amine group and what? Carboxylic acid group. Okay, that's why it's called amino acids. All of them. So now there are around what? 20 different amino acids. So what's different? The side chain. This is called a side chain group, guys. R group. That's what's different for all of them. So one of the things that we talked about, guys, for some of the folks here, guys, that we already discussed. This side chain group, okay? You have to know, for AP Bio, in my understanding, you have to know one of them, okay? You gotta know one for AP Bio, okay? That's called cysteine. You have to know this guy. Cysteine, guys, is one of the amino acids that you got for AP Bio. You might want to know what it is. You can see other stuff. Like, I mean, you might want to know their names. You see them, you hear them, you should know what it is. Okay, proline, like cysteine, leucine, glycine, like glutamic acid, like glutamin. They're all like, all of them is amino acid. Okay, you should. But I think, okay, one thing unique about the amino acid, you have to know this guy. For API. For some of the other folks, you gotta know all of them. Okay, like you guys have to know every single one. Okay, you gotta know each and one of them. You have to know what their polarity is and all that stuff. And their symbols, like cis, becomes a cysteine of the amino acid, like abbreviations as well. But yeah, but the difference is the R group. Okay, that's what the difference is. Okay, so what makes them leucine, glycine, whatever, guys? It's that R group. Okay, so based on this R group, okay, the amino acid can be classified as hydrophobic, okay, or hydrophilic, okay, or it can be also called plus charge or minus charge, okay. They have a plus charge amino acid, minus charge amino acid. The plus charge is basic amino acids, which typically they contain nitrogen. And this is what, guys? Minus is what? Acidic, which they contain carboxyl group on the side chains. Okay, on the side chains. So there are like maybe 4 classes of amino acids: hydrophobic, hydrophilic, basic, and acidic. Okay, there are 4 classes of the amino acid that you guys have to know. Okay. And again, like, you know, hydrophobic could be like glycine, for example, is like hydrophobic. Okay. Glutamine might be hydrophilic. Okay. Like basic is I don't know, like leucine. I'm not sure. Acidic is glutamic acid, for example. So there are some of the amino acids that falls into these 4 categories of the amino acid based on the side chains. Okay. That's important. But what defines which amino acid it is what? Based on the side chains, because they all have the same thing. Amino acids be all the same, except the structurally, I think proline might be a little different. This might be a little bit different in terms of structure-wise. I think the side chain has a little bond made with the the nitrogen group here. But that's only difference, guys, structurally. But all of them should have the same like basic shape as amino acids. Okay. So what is the primary structure of the protein, guys? Is what? Sequence of the amino acid sequence. Okay, which one comes first? Oh yeah, you know, for a protein, this one comes first, second, and third, like the sequence, right? That's what we call the primary structure of the protein sequence. Okay, so yeah, methionine, like the other guy, leucine, glycine, like glutamic acid, like whatever. Okay, but sequence becomes what we call the primary structure of the protein. Okay, and now where do they get the primary structure from? Where do they get it from, guys? Is what? It's coming from DNA. So your body should produce a specific enzymes, right? Like, yeah, dude, like, you gotta understand, guys. Okay, your body should produce like certain enzymes, right? So, for example, like what, guys? So for example, guys, your body should produce, you know, stomach acid, stomach enzymes, right? Which is what? Pepsinogens, right? So your body, when you guys eat food, pepsinogens should be produced, okay? Or is there, and then they should be activated to pepsin or something, okay? So it's an enzyme, okay? That's an enzyme. So how does your body know how to produce it? Your cells are producing it, okay? Where do they get that from? The sequence, right? They have one job. You have to keep in mind. The pepsin, which is the enzyme in your stomach, they have one job, which is what? Break down protein, okay? They got one job. If you guys eat steak, their job is what? To break them down, okay? And to break it down, what happens? They have a very specific shape, okay? So where do they get that shape from? Okay, so how do they know how to produce that? So that sequence should be all the same. Every time the sequence is all the same, okay? So you know, when you guys go look at your like protein, they have all the same sequence with the pepsin, okay? Where do they get that from? Dude, it's not like randomly, okay? Oh, today, this guy first, tomorrow, this other guy first. No, it's very structured, okay? Where do they get it from? Your DNA, okay? So your DNA has all that sequence decoded in, okay? And then what happens, guys? It turns into the primary structure of the protein, okay? Keep in mind, guys, one of the stuff that we talked about is what? DNA should not change, okay? You do not want your DNA to change, okay? A lot of times, if your cell is acting normally, if the DNA change has been observed, they try to fix it, okay? They go in and try to fix it, or what? If it's terminal, what happens, guys? Yeah, they kill off the cell, okay? So typically, that's what happens, guys. So DNA is very important, guys, because what? They have what? Like primary skirt for what? Primary structure of the protein. Just so you know, okay, which is very important for the shape of the enzyme overall, okay, because they got the sequence, okay. So let's talk about, guys, the secondary structure of the protein. So first thing first, guys, one thing to note: the secondary structure is basically, guys, so I'll be very like, you know, whatever. So the secondary structure is basically alpha helix or beta, okay, like they call pleated sheets. Okay, this is basically, guys, the secondary structure, okay? So what happens is amino acid chains up. Okay, first thing I'm saying, guys, how do they chain up? How do they, guys, like chain up with the other guys? Same thing, dehydration synthesis, right? So, guys, basically what happens is what? There's a H group here, guys, and there's what? Another amino acid. There's one more. Do you see that, guys? Like, there's one more, right? I mean, at the moment, guys, you guys should know what really happens here. What happens? How do they chain up, guys? Dehydration synthesis again, right? As water pops out, okay, what happens, guys? There's a bond created. Right here, like this, okay? And now, what do we call this bond, guys? We call this what, guys? We call this peptide bond, exactly. peptide bond, okay? Based on what? Dehydration synthesis, okay? Keep that in mind. So now, yeah, so going a little further, guys, okay? What do you think this, I mean, so, as in terms of functional groups, okay, teachers don't want to go this far. When you see this, okay, when you guys see this, there's a carbon with double bonds, okay, and there's a, if you look at, there's a carbon here, there's a nitrogen there, okay? R is a carbon chain, okay, like carbon here. Yeah, what do we call this, okay? So, yeah, what kind of functional group is this? Anyone know, guys? So, if you look at it, guys, this is carboxylic acid, right? This functional group corresponds to the carboxyl group, right? What do we call this? Anyone know, guys? Functional groups? Yeah, what do you think? Functional, yeah, what do you think? What do you think?
Well, yeah. You don't know that.
Huh?
No.
So what do you think? Okay, anyone?
Like, check my notes.
That's it. This is called the amide. Amide. Okay, that's called the amide, guys. That's a functional group, guys. When you see this with the nitrogen there, okay, it's called the amide. So I mean, it's good to know. Okay, good to know. Okay, functional group wise. I guess you guys. I mean, your teacher may walk you through the functional groups, guys. So you gotta know the amides, you gotta know the amines, carboxylic acid, aldehyde, ketone, and all that stuff, guys. Okay. So that's all in the reading, guys. I'm not gonna go there. Okay, you just have to memorize which one is it, what is what, guys. Okay. So they make a long tail of that guy, okay? Long tail of these guys, okay? So based on the side chains, like what it is, you know, it makes up what, guys? A primary structure, right? But now, every amino acid, guys, as we know, so they create, guys, like chain of long amino acid, okay? So keep in mind, guys, here, this side, I think in bio, they don't really call this like two whatever, guys. On this side, it's called the N-terminus, okay? N-terminal. So the last amino acid on this side is called the N-terminal, okay? Because why? Like nitrogen. This side is what we call the C-terminal. Okay, the last carboxyl group here, okay? Again, there's a long tail of these guys. So they chained up, yeah? It's a primary structure, guys. What about secondary? What the hell is that? Like, what do they do? So it's called the alpha helix, okay? So it turns into like a helical shape, like this. Okay. Most of all complex proteins have an alpha helical shape, okay? And now, basically, you got to understand, guys. I mean, where does this come from? It's come from what, guys? The main thing that produces them is what? Hydrogen bonding. You gotta know that, okay? So I'm not here to discuss hydrogen bonding again, how they produce it, okay? You gotta understand, guys. So hydrogen bonding is what, guys? The oxygen, okay, which is this guy right there, okay? On the carboxyl group, okay? And also what, guys? The hydrogen on what? The amine group, okay? What happens, guys? This hydrogen is eligible for hydrogen bonding, okay? And they are creating the hydrogen bonding, okay? And that is what? What creates the alpha helical shape, okay? Keep in mind, yeah, these hydrogens, right? These guys, right? And what? These oxygens, okay? If they're attracting, that's what creates hydrogen bonding, okay? And what happens, guys? That attraction is making them into what? Alpha helical shape like this, okay? Make sure you understand what I'm saying. Some small, tiny amino acids, or proteins, they might not even have this, okay? But a lot of times long, big the proteins, they all have the alpha helical shapes, okay? Or the secondary structure. This is called what we call the secondary structure of the protein, okay? And then also, guys, the beta sheets, okay? So again, like, there's no, like, one of the things we have is, I'm saying, guys, the term alpha and beta, guys, and there's not much significance there because they found the helical shape first. That's why we call it the alpha, okay? And the beta sheet was found the second. That's why they call it beta, okay? There's no like special meaning in that beta and alpha. I'll just let you know. And now, one thing to note, guys, here for the beta. If you look at it, guys, so they create like a little sheets like this, okay? Like a little, like piece of paper sheet, okay? And one thing to understand is what, guys? So that also is due to also hydrogen bonding as well. So the carbon with the oxygen there, okay, with the hydrogen with nitrogen, okay? So there's attraction between these guys, hydrogen bonding. So they're making that throughout the entire sheet, okay? As they run, as these amino acids run in parallel like this, okay? What happens, guys? They make hydrogen bonding, and what? They make it into what? Little sheets of the amino acids, okay? So these alpha helix shape and these beta sheet guys, they can be in the same single protein, okay? And that's what they call, guys, secondary structure of the protein, okay? So keep in mind, guys, the alpha helical shape and beta sheet is not unique to a particular protein, okay? Most of them have it, just so you know, okay? So it's not like, oh yeah, this protein should have this, the other protein doesn't, okay? It's not a uniqueness. But one thing to note, okay? Proline, okay? So this guy, just so you know, proline. If you take a look at the structure of the proline, okay, so I'm not like 100% with the, I don't memorize the structure of the amino acids just too much, guys. Prolines look a little bit interesting, guys, okay, because they like have this ring or something, okay, and it makes a bond of nitrogen there. So that's like what proline looks like. Keep that in mind, okay, you go and look at the document that I gave you, okay. Proline, okay, so when they create like some structure like this, okay, so they create, okay, some kind of like, you know, angle, like a little different pathway when they turn into, when they try to create like secondary or tertiary structure, okay? So they give that little unique curve in the amino acid. I mean, again, not for AP, it's fine, okay? But one thing to note, guys, the structure of the proline is a little bit different than the other ones, okay? So guys, I think the most important thing of all, guys, is this guy, tertiary structure. This is one of the most important things, by the way. Tertiary structure, guys. Definitely have to know this one. And a lot of questions is on this guy. A lot of the AP questions that FRQs, guys, is based on tertiary structure proteins, okay? So, tertiary structure proteins, now you guys have the primary and secondary structure of the proteins. What's a tertiary? So tertiary structure is where a proper, unique, okay, shape of the protein is being shaped up, okay? So this is where, guys, where you probably heard the term protein folding comes in, okay? So they got chained up and they got primary and secondary structure of the protein all kind of nice lined up, right? So but which is not unique to any particular protein overall, okay? What happens, guys? Okay, this is what really gives them the uniqueness of the protein overall, okay? So we talked about it, guys. A lot of proteins are globular protein, which act as an enzyme. Okay, enzyme must be unique. Okay, they should only catalyze a specific reaction, right? Just we talked about, right? And where to get the uniqueness, guys? Protein folding. First thing first, guys. Where does this occur? Where? Okay, so protein folding. Okay, so unless you guys live on the rock here, guys, they get the protein. Okay, gets like translated in what? Ribosome. Okay, so where does this occur? Protein folding. So protein folding occurs, okay, in the something we call the chaperonin. Chaperonin. It's in the text. It's in the reading, guys. Okay, or chaperone protein. Okay, chaperonin. This is important. Okay, they even asked us to say this. They even asked us on the SAT too. Chaperonin, like a chaperones. Okay, chaperone protein. The chaperone protein, okay, so this is a little protein capsule, okay, so it's like a little area, okay, where after the protein has been created, they go in here and complete their protein folding, which gives them the unique shape. Why? Yeah, we have to ask yourself, guys, what the hell, why? Why do they go into a little space, okay, where it doesn't really get impacted by other stuff, and then have to do it in here? Because the importance is, guys, okay, you need to, in order for the enzyme or the protein to function in a specific way, they have to fold, okay, they have to like fold in a unique shape, okay? That's a very important process of the protein synthesis, okay? So they go into, okay, so I mean like within the cytoplasm, okay, think of the cytoplasm, right? it's got a lot of what? Dude, it can be acidic, it can be basic, it can be like, there's a lot of stuff in there, guy. It could be like, dude, there's some other enzymes that might alter the protein folding. So instead of that, what happens, guys? Like, just like, oh, dude, you guys have important tests coming. What do you guys do? You guys go into a little small room. There's no computer. You know, depends on, like, there's nothing else in there. No, you don't have your brothers, like, you know, sisters and siblings, whatever. You go into a small tiny room and what? Yeah, like you study there or take a test there, right? Same thing. It's a very serious process. It's a very important process. So that what? They have a space here where they go in and what? And complete their folding process. Okay, that's called chaperonin or chaperone protein. Okay, so make sure you understand, guys. You guys have to know this for AP, Olympiad, SAT, Mac, and Time. Okay. So what the hell is tertiary structure then? Okay. Make sure. This is very important, guys. Okay. So this guy, okay. So this is like hard to draw, guys, by the way, okay? So what happens here is, once they complete their secondary structure, okay, they have to fold, okay? You know, they have to fold in a specific way, unique way, okay? And that uniqueness, okay? Again, uniqueness is what, guys? So if you look at it in terms of the amino acids, they have what, guys? These side chains. Okay, these side chain groups, right? So now what happens to what, guys? Okay, so this guy is what? Hydrophobic. And this guy is hydrophilic. Okay, and this guy is acidic. So what will occur in terms of the folding process is what? So again, if this is a protein overall, I'm sorry, guys, like my drawing, I can't really draw that well, okay? All the amino acids, okay, side chain groups, that is hydrophobic. Yeah, so keep in mind, guys, okay, outside is what? Outside is water. Okay? Yeah, what do you think will happen to the hydrophobic amino acids? They're gonna congregate in the middle. Okay? They're gonna go all in the middle where the water is not present. You understand, guys? So these hydrophobic amino acids will be like all in here. Okay? Yeah, what do we call that, guys? So all the amino acids, that's what, like hydrophobic, like, you know, side chains, what, they're all in there. Okay? So they fold, and what, they go in there. Do you understand, guys? Okay? So now, this is what we call nothing special, what? Phobic interaction. Okay? So the hydrophobic side chain amino acid is what? They're all found in what? In the middle of the amino acids. You understand, guys? Yeah, what about guys like these acidic or basic amino acids? What do you think, guys? Or even hydrophilic? They're all what? On the outside. You understand, guys? That's where they position themselves. You understand, guys, here? So that's like the overall, like very high level looking at it, okay? So on the outside of the protein is what? You find philic and what? Plus or minus amino acids, okay? And within this like protein overall, okay, and this is what, like, dude, there's hydrophilic interaction, like plus or minus ionic interaction as well. Do you have a question?
Yeah. Is it like outside the protein, or are you just saying it's like near
like on the
outside?
outer portion of the protein, okay,
Right,
where it faces the water.
Not actually outside the protein.
Yeah. So it's outer surface of the protein per se, okay. And also keep in mind, guys, you know, so like within here, like folding, okay, like within here, just so you know, okay. So there could be what? So this protein shaping can be happening due to what? Like, okay, it could be due to hydrogen bonding. So they asked this quite a few times, hydrogen bonding. So what is you know, responsible? Like what bond are there, right? So we know the secondary structure is what? Based on the hydrogen bonding of the hydrogen bonding of what, guys? The carboxyl and the amine group, right? Tertiary structure, what, guys? A lot of things, okay? Hydrogen bonding, okay? Sometimes they call it what? Also hydrophobic interaction. So that's what, guys? Like, yeah, the congregation of the hydrophobic molecules in the middle, okay? So all these like hydrophobic amino acids, they all like accumulate in the middle, okay? And also what? Ionic interactions. Okay, these plus and minus charged ions or amino acid, they kind of what? Chain up, yeah. I would just. So I'm sorry. It's what? It's ionic interaction. Okay. So you gotta write this. This is the most important thing of this entire chapter. Okay, because if there's a question on this, guys, a lot of times this is what they ask you. Okay, ionic interactions. okay, between what? The plus and minus charge amino acids. The side chains, like these are groups, right? The side chain groups, right? And now keep in mind, guys, every protein has a unique side chain sequence, okay? Like if you have two different proteins, they have what? Different sequence, which is they have different side chains, okay? Sequence-wise. So they have what? Fold into a different protein like this, okay? And also what, guys? And also they can create, as well, also what, guys? Covalent bond. 요게, I think you have to know this one, okay? Covalent bond. So in terms of folding process, guys, okay, so they all occur like in this guy, the chaperone, okay? They folded in there. And now covalent bonds. So you got when they ask you, oh, by the way, tell me about how can a protein folding occurs in a unique pattern. They always ask that question, okay? So yeah, hydrogen bonding, hydrophobic interaction, ionic interaction, okay, hydrogen bonding, you know, and also what? Covalent bond shapes up the uniqueness of the protein. That's how you say it, okay? So where do they get this covalent bond? Covalent bond does not really occur too much on like tertiary structure of the proteins, by the way. Even for the quaternary, the covalent bond doesn't really happen. But now, the covalent bond occurs as one thing that I mentioned about the amino acids, but you gotta know one of them, right? Cysteine. You have to know this guy. Cysteine amino acids, they can create something we call disulfide bridge, okay, which is a covalent bond, okay. I tell you guys, okay, if you write this on your AP exam, like, oh yeah, like, you know, they will ask you a tertiary structure protein, okay, blah blah blah, answer this question for me, okay. If you mention this guy and this, which result in covalent bonding, which shapes up into a certain shape of the tertiary structure of the protein, they can't mark it wrong, okay. You do not give them a reason to get it wrong, okay. And this one is very important. It's cysteine, you know, like disulfide bridge. So cysteine, along with methionine, they are the only amino acid with sulfur in it. Okay, so you know sulfur. So these amino acids, guys, they have like amino acids. What do they have, guys? They have a carbon, they have hydrogen, oxygen, and what? Nitrogen, right? But two of them have sulfur in there. That's it. You get two of them. So if you look at it, right, cysteine, they can create. So if there's a two cysteine amino acid, okay, cysteine amino acid, guys, here, what happens? They can create what? Disulfide bridge with covalent bonding. Okay, covalent bond is what, guys? Very strong compared to what? Any one of these guys, according to bio. According to bio, okay. What's wrong with hydrogen bonding, hydrophobic interactions, ionic interaction, and stuff like that, okay? So the cysteine, okay, this stabilizes. Then they make the what? Tertiary structure of the protein stronger, okay? So yeah, so it's something that you might want to mention on your test, guys, by the way. Disulfide bridge, okay, based within what? Cysteine, you know, amino acids, okay? That's one of the things you might want to mention. And that shapes up what? Specific shape of the proteins. Make sure you understand, okay? Tertiary structures. So, and then also, guys, again, since the time, I need the time to explain the next one. So, quaternary structures, okay? So quaternary structure, to explain the quaternary, the fourth shape of the protein, right? The best way that a lot of times that the College Board will bring this up, quaternary structure, guys, is a hemoglobin. That's when they bring this up, hemoglobin. Hemoglobin is what, guys? It's one of the carrier for the oxygen gas in the red blood cells, okay? So hemoglobin has two alpha subunits, okay? These are all like protein, like tertiary structured proteins, okay? Two of the same ones, and also what? Two of the beta shapes. Beta subunit, alpha subunit, there are four of them, okay? And now when there are multiple tertiary structured proteins come into one single larger subunit, okay? And this is what we call, guys, what? A tertiary structure, quaternary structured proteins overall, okay? And keep in mind, guys, interaction between these guys, okay? Like how they interact is what, guys? All what? Either ionic interaction or what? Like hydrophobic, hydrophilic interactions, okay? There is no covalent bond between these guys. Okay, that's one thing to know, guys. Okay, so AP, like I don't know, you guys probably don't want to go that far. Just let you know, okay? But again,
interaction between like quaternary structures, what guys? Yeah, the ionic hydrogen bonding, hydrophilic interaction, blah blah blah, guys. That is what really makes them what? Kind of attach them together as a quaternary structure, as interaction between these what? Subunits of the tertiary structure protein as a quaternary. Okay, that's one thing to know, guys. Okay, as a quaternary structure proteins. And this is something that you have to make sure you understand, guys, because this is one thing to note that I'm going to bring it up again as for the allosteric, okay, cooperativity or inhibition. Okay, I'm going to bring this up, okay, today, like right after this, by the way. So that's what gives them, okay. So these, like, you know, the very uniqueness is what giving them what, guys? Specific, unique structure as a protein overall. Make sure you understand what I'm saying. And also, guys, because of there's so many bonds involved, hydrophobic interaction, philic interaction, ionic, blah blah blah, okay, like cysteine, like disulfide bridges, and all this. There's so many bonds. Do you understand, guys? That creates this entire protein overall. They're what, guys? They're prone to what? Break when what? Condition is not right. Okay, and that condition is what, guys? Either what? Temperature or pH, exactly. So, I mean, dude, you guys all know, right? If you increase temperature, what happens, guys? Yeah, temperature, there's more energy. These guys move faster, okay? Like chemistry, right? Yeah, temperature goes up, what happens, guys? Oh yeah, they move what? The average kinetic energy increases. What does it mean, guys? Every single of the molecules, or even the atoms, they move faster. If they move faster, what happens, guys? They break. The bonds are breaking, do you understand, guys? Okay? So, if they break, what happens, guys? They lose their specificity, okay? They're supposed to react, okay? A certain reaction, a catalyze certain reaction. It's not gonna happen because what? It becomes what? Different shape, okay? Perfect example is what, guys? I mean, so this is something that you will have to nail for the AP level as well, okay? What is the most abundant enzyme in the surface of the earth? Do you guys know, guys? Yeah, what do you guys know? Most, like, you know, abundant protein or enzyme. What do you guys know? It's called rubisco. Rubisco. Where do you find this, guys? In every plant, you find this. Every plant. Okay? What's their job, guys? Fixes or bring in a carbon dioxide. Okay? They have attraction for carbon dioxide or CO2 molecules. Okay? If plants, temperature goes up a little bit. Okay? Temperature goes, yeah, maybe today, you know, 100 degrees Celsius, 100 degrees Fahrenheit. Okay? Maybe tomorrow, 110, 120. Okay? Like Arizona and Las Vegas, right? Guys, if you go to Las Vegas, right? Do you see a lot of plants, guys? Yeah, do you see like the plants we see here, guys? You don't, because why? They die because why? When temperature gets hot, what happens, guys? Rubisco, it changes the configuration just slightly, okay? It's just like, you know, and the enzyme shape changes just a little bit, okay? Which means what? They fail to bring a CO2 in. So temperature, because temperature, guys, okay, can change the bonds, it's very sensitive. The enzymes are very sensitive to temperatures, okay? So keep in mind, guys, just so you know, one thing you gotta know this one as well, guys, when the chapter comes, guys, for those photosynthesis. So temperature, and again, like if temperature is too high, like if it goes above like, I don't know, like 40 degrees Celsius, for example, guys, it might change the configuration of the protein a little bit and goes back to what it was, okay? So it might not be like irreversible, but at the same time, it can go back to what it was. Temperature goes above like, you know, 100 degrees Celsius. What happens, guys? Sometimes they will break and what? It becomes irreversible. It becomes a different protein overall, okay? Like what is the example, guys? Like when you guys cook meat, what happens, guys? Can you go back to what it was? Can you make it raw again? You can't, because why? You already changed. You already denatured the protein. Do you understand what I'm saying? So term denaturation is what you need to know. Okay, well, you know how to spell, right? Denaturation, okay? That's one of the things you have to understand from the temperature perspective, okay? You have to know, that's like, dude, like you probably learned this in 7th grade, 8th grade, whatever, okay? And pH is what, guys? We talked about pH, guys, last week, is what? Okay, that's concentration of H+. Okay? So, you gotta understand, guys, when there is additional H+ or additional like OH-, for example, guys, there's a greater amount of H+, what happens, guys? We talked about it overall, the outer portion of it. Okay, so if, for example, guys, we have plus charged amino acids, we have minus charged amino acid. If there's a sudden influx of H+, what happens, guys? What do you think will happen? The plus charged ones will be what? Moving away. Okay, do you understand what I'm saying? Okay, which means what? Yeah, they can change the configuration of this as well. Okay, so there's an impact here because what? There are amino acids that are what? Plus charges and minus charges. Okay, which means what? Yeah, they repel by the same charges. Okay, they're attracted by the different charges. Okay, which will change the configuration, which will also what? Further can denature protein as well. Okay, you can activate it, but you can also denature the protein if it's too much. You have to understand. Okay, so one like obvious thing is what? Denaturation of the protein based on what? Temperature and pH. Very obvious stuff, guys. Nothing fancy, you know, to understand this one. Okay. And sometimes, depends on the degree of it, it can be reversible. Sometimes what? It's irreversible, okay, if it went to a certain degree. Any questions, guys? Any questions or something else? Okay. So, with all this, guys, so I have to, you know, go into, guys, now, like, moving into the enzymes, okay, and then, the spontaneity stuff, okay? The mechanisms of the reactions and stuff like that. So I got to talk about that, guys, a little bit. So I think I can finish that all by today, okay? And the more important thing of that aspect, guys, is where you guys have to understand the enzyme like reactions, inhibitions, and allosteric like cooperativity and inhibitions. That's the most important thing of this, and that's like feedback system and all. It's pretty short, but that piece is what you need to know to answer the question of the homework, okay? Which is the side. So, okay, so, and this thing is recorded, guys. By the way, this is not for like people like like who didn't show up today because some of them told me they can't make it on some of the days or something. So, and for you guys to go back and review, okay? So, I'll share the recordings with you guys, and it's already uploaded, guys. Okay? So you guys can go in and review it if you want to. All right. So, so now the chapter, guys, that I'm talking about. I'm gonna be very quick with this. I mean, because AD Bio, they don't go too much with this, guys. They don't go too crazy on this chapter, okay? So, first thing first, guys, okay? Let's talk about, guys, a typical reaction in bio, okay? So, and we'll talk about, guys, why reactions require enzyme or even like ATP, for example, right? So let's talk about, guys, a typical reaction, okay? Sometimes, guys, I mean, they use a term in bio, free energy, okay? They use a term free energy, okay? So they don't use a term like kinetic. I mean, kinetic, I mean, depends on which bio you're talking, guys. They like to use the term, guys, free energy a lot, okay? Which is what? G, okay? If you're coming from chem or something, G, free energy values. So, as you guys all know, right? I mean, if you guys know. If for a reaction, like A turns into B, okay? So one thing to understand, guys, for the reactions to occur, okay, so let's say, for example, in terms of free energy perspective, I'm not going to define what free energy is, guys. Free energy is what, guys? I mean, what's free energy, guys? Yeah, what's free energy? Indeed? Indeed?
The amount of energy in the system.
Yeah, what do you think? What's free energy?
Energy that can be used in a reaction.
Thermal energy.
Huh?
Thermal energy.
Anyone, guys? Free energy, guys? Free energy? Okay. Free energy is what? So free energy is how much energy that's stored within a molecule, okay, that can what? Able to perform work, okay, that can what? Like put into work. Okay, like again, like okay, so then we can go into okay, what is work? I'm not interested in that, okay? So free energy is not potential energy, by the way, because they're not the same thing. Okay, potential energy is how much energy is stored overall, like how much energy, how much money do you just have in your bank account, okay? I have like $100,000, okay, in my bank account. But free energy is how much can you spend, okay, on your car, the next car, like I'm not gonna spend $100,000 unless like, you're like, those one of those people, right? So something like that. Free energy is how much energy you can put into work, put into what? Work for a reaction, okay? So now, as we know, delta G, okay, make sure you understand, okay? Delta means what, guys? Not change is what? Free energy of the product minus what? Free energy of the reactant, okay? Product minus the reactant, okay? So, typically, right, if delta G is positive, which means what, okay? Which means what the product has more free energy. Okay, like, so what does it mean, guys? Reaction perspective, that's what it looks like. This is a free energy here. This is a reactant, that's a product. Okay? So which means what? The product has more energy compared to the reactant. Do you understand what I'm saying? Okay? And that's what we call delta G as what? Positive reaction. And in bio, what do we call those guys? So there's a special name for this. I mean, so in chemistry, we call this what? Non-spontaneous reaction. You gotta notice, non-spontaneous reaction. Okay? In bio, what do we call this, guys? Endergonic reaction. Okay, it's called endergonic. Okay? And you gotta know this one, guys, okay? For AP bio. Endergonic reaction, okay? Which is delta G is positive. And in chemistry, we call this what, guys? Non-spontaneous. Okay? Which means what? The reaction will not occur on its own, okay, in a very high level. So, okay, how can this reaction happen? Like, how do you make this reaction happen? I mean, so, because you got, dude, like, you gotta put in this much work in order for the reaction to occur. Okay, how can this kind of reaction can happen on its own? Okay, like in your cell, this reaction must happen. Okay, I mean, like, okay, how? Like, endergonic means what, guys? It doesn't happen on its own. Okay, you have to have what? Other, such as what? A coupled reaction. Which means what, guys? There must be other reaction that goes along with this guy to make this happen. Okay? So, such as what, guys? Such as, okay, so with this guy, I'm sure you probably heard this like many times, okay? What is considered as the energy within the cell, guys? The currency of energy in the cell, guys? ATP, okay? Adenosine, we talked about it last week, right? Adenosine triphosphate, okay? ATP, right? So, okay, so because this reaction is not gonna happen on its own, we're gonna give them what? ATP. I'm sure you heard this many times, right? ATP, right? So ATP turns into what, guys? Turns into ADP and phosphate and provide the energy for this reaction. So overall, this reaction can occur. Okay, so how the hell does it happen, right? So okay, so so you gotta understand now for DP bio. So SAT bio, regular bio, guys. It wasn't a big deal for you guys. Oh yeah, ATP energy, guys. Energy, here you go. This can happen now. Oh, next chapter. No, like not anymore. Okay, you gotta know a little more now. Okay, so you gotta understand first of all, how can ATP give them the energy? Okay, but this guy turns that guy. How can this provide them the energy? Something is now. Okay, so a lot of times bio teachers, guys, they make a mistake with this. Okay, so let's talk about the ATP now. So ATP, okay, well, adenosine. We talked about it, guys. Adenosine triphosphate. We talked about it, right? So guys, we talked about it, guys. Is what? There's a adenine group here, okay, nitrogen and stuff like that, okay, and also this is a ribose. Carbon here and phosphate. Okay, so if you look at it, guys, here to here is what we call the adenosine. We talked about it, guys. Okay, and it's got what? 3 phosphates on it. Okay, it's got 1, 2, 3. That's so we all know, guys. What this has a lot of energy. Okay, but what energy are we talking about? Is what this refers to? What free energy. It has what high free energy. They can do a lot of work. Okay. And what happens, guys? What the hell happens now? So this is where, I mean, people know this. Okay, what's the problem, guys? When this bond breaks, energy releases. So the misconception a lot of students will have as well. When bond breaks, any bond breaks, energy releases. That's a misconception for a lot of students, by the way, because of this. That's not true, guys, okay? So as you guys should know, guys, from chemistry perspective, when you're trying to break a bond, what happens, guys? Energy must be put in. Energy must be going in what? Energy must be put in for this process, okay? So when you break a bond, energy doesn't come out. It's what? Energy is what? Being put in, stealed away, okay? But how can this provide the energy? Keep in mind, guys, okay? ATP turning into an ADP and phosphate, okay? Like this is breaking. This guy comes off, okay? Why this can give energy what, guys? This process what, guys? This process energy releases, and what? This becomes what? Delta G is what? It's negative. Okay, this is exergonic process, okay, which means what? If you look at the diagram, this is ATP, okay, and ADP and phosphate is what, guys? It's on the bottom. Okay, it has what? This is more stable, less energy compared to that guy. Okay, so this is what happens, like this. You see that, guys? So which means that this much energy is what? Released, okay? They got less energy, okay? So you guys should know, what do we call this process? This is what? This is what we call energy of activation, okay? Energy of activation. which is what? Amount of energy needed for this process to occur. Which means what? This is the amount of energy needed for what? You need to break this bond, okay? And if that breaks, what happens, guys? Yeah, this is what happens, okay? And then one thing you have to understand, guys, why do we use ATP? Okay, why ATP? Why not? Can you get it from straight from the glucose? Because why? This bond is what? Relatively easy to break. Okay, so they call this what, guys? Like currency of energy within the cells. Okay, so like, dude, if I want to go buy something in a store, okay, like, dude, I know my phone, right? I don't know how much it's worth now. Okay, maybe I don't know, like $500. Okay, I'm not sure what this. I mean, I'm not sure how much it's worth now. If I go to a store, guys, like Best Buy, okay, I want to get a PlayStation 5, okay, can I give them this and I want PS5? I mean, they don't even have it, but are they going to give it to me, guys? No, you have to liquidate this. Turn it into what? Turn it into money, okay, and what? They're going to give the PS5 if they have it, right? So, same thing, okay? They have to change glucose into what? ATP. Why? This is one of the ways they can what? Provide the energy, okay? And now, why do we use this? Why do we use $1? Because why? Dude, they accept it everywhere, okay? Easy to break, okay? The first, second, and third, okay? This is a very easily broken, okay? This guy, guys, just so you know, you can easily break this guy, so it's relatively what? Easier to use them, okay? Which means what? It has what? Low energy of activation, okay? So why? I'm not gonna talk about it, okay? So how can this assist, okay, for A in terms of B reactions to occur, okay? This is how. So you look at this, when you couple it with the ATP, what happens, guys? So this is what, for example, guys, a delta G-wise of the reaction. This is negative 100 kilojoules per mole, okay? This goes out, it's plus 50 kilojoules per mole. This reaction requires 50 kilojoules. This one gives you 100 kilojoules, right? Overall, what happens, guys? Overall reaction becomes what, guys? Negative 50 kilojoules, which is what? Delta G is negative, which makes it what, guys? Overall reaction becomes what? A spontaneous reaction. This is Hess's law, okay? You don't have to go crazy here. So by using ATP, okay, and coupling it with this, okay, this is what? This becomes positive, which is what? Non-spontaneous. It doesn't happen on its own, okay? But by giving them this, what happens, guys? Overall reaction becomes minus 50, okay? Which is a spontaneous process. Do you understand, guys? So that's the overall understanding how ATP assists, okay, the overall reaction within the cell. But you gotta know a little more than this, guys. So, okay, how in detail this occurs, okay? So what the hell is really happening? Okay, we understand from the higher end perspective, okay, high-end perspective from the outside. Yeah, we understand what is happening overall from the math perspective, but what the hell really is going on? So what really is happening is this. This is something you have to know, okay? Because of the reason is, guys, you have to understand something called kinase reactions, okay? There's an enzyme that does it for them. There's an enzyme that runs a specific reaction like this, okay, where they put a phosphate group on like something, okay? They use ATP and make this kind of reaction occur, okay? That enzyme is called a kinase. So what the hell is going on? So what's going on here is step by step, right? Yeah, A is going to react with ATP, okay? And what's going on, guys? They are phosphorylating. The phosphate group, right? One of the phosphate group that came out, they attach it onto this guy. Okay? And what? ADP. Okay? So what's happening is, guys, what happens? This is A, right? And this is ATP, right? They got 3 phosphate group on them, okay? And then they have a ribose and whatever, okay? So they put this guy in here, okay, onto A. Okay, so now what happens, guys? A has what? A phosphate group attached, okay? And this kind of reaction is what? Catalyzed by something called kinase. You gotta know kinase is absolutely needed for your AT bio, okay? And it's something we'll discuss later down the road. A lot, guys. So what happens, guys? So now, when you phosphorylate something, what happens, guys? Their free energy increases, okay? So now, yeah, look at this, okay? This is A, that's B, in terms of the free energy perspective. This is A and B. When you phosphorylate A, what happens, guys? It's now above. This is A phosphorylated. You understand, guys? Okay? And then what happens? A turns into B, becomes what, guys? Now, an exergonic reaction. Do you understand, guys? Something you gotta know. Okay, your teacher will definitely bring it up. And that's basically, guys, how this occurs. Okay? So now you should know, okay, how can ATP really assist at a molecular level, okay, how things can happen as well. Yeah, dude, there's an enzyme such as kinase that take the phosphate out from the ATP, puts on this guy, which increases their what? Free energy, where the reaction now can occur, what? Exergonic pathway, okay? I mean, like, this is even this is a high level, okay? Which means, like, you know, in the fall semester when you guys like go to school and whatever, guys, your teacher can hopefully explain this a little more in detail, okay? But this is relatively how things can occur within a cell using ATP, by the way, okay? So spontaneous perspective, thermodynamics perspective, okay? This is how the reaction can occur, okay? This is these are what we call what we call this guys what? A coupled reaction. Okay, two things occur, you know, together, you know, coupled reaction. Any questions, guys? Yeah. Questions, concerns? So where would you hear this a lot, guys? Kinase. So there's a chapter, guys, which it's called a cell communication chapter. Okay. So like one of the kind of example guys I got that where this comes up a lot. These things come up a lot, guys. Is where I'm gonna do it over there. Is where guys. They call this guy. This is very important, by the way. That's why I'm like mentioning this. Okay. It's called cascade of phosphorylation. Okay, that's what it is. Yeah, cascade of phosphorylation. So that is something we'll discuss. I mean, this is definitely the summer. Okay, we'll discuss, guys, in the summer. I don't know when exactly, towards the mid portion or towards the end. Okay, cascade of phosphorylation. So what this is, okay? For example, guys, right? If there's an insulin, okay, and this is a receptor protein or whatever, okay? So if this gets activated, okay? So what is the role of the insulin, guys, from the cell? What's the role of insulin, guys? To make sure what? They're telling cells to what? Okay, absorb the glucose in the blood. That's their job, right? Like, oh yeah, their insulin is telling the cell, okay, like, dude, absorb the glucose. What do they do, guys? They either have to activate the channel to let the glucose come in. Okay? Glucose is a polar molecule, which means what? They can't just come into diffusion. The channel must open. Okay? Either they have to produce this channel, okay, protein synthesis, right? Or what? If their channel's already there, they open that guy. Okay? So which means what? It's a mechanism, right? So to get this message they got from insulin, okay, all the way here, they have to what? Go into nucleus. The nucleus will tell them what to do. Okay, either activation or deactivation or production of a protein. Okay, so you know. So then, so this pathway from the insulin to receptor with the insulin to the nucleus, that's where the cascade of phosphorylation occurs. Okay, which is what? This thing is happening in many steps. Okay, cascades. So that's where these things will come up again. Okay, which means you cannot forget, you know, the stuff we just talked about. Bio, man, I tell you, it's a very like you gotta remember stuff, okay? Like you can't. But chemistry, right? Like you learn, like you forget like bio. You can't do that. Okay? You gotta remember your stuff so that because it'll be applied to the next chapter. Okay? I mean, bio, believe it or not, is not very cumulative. Okay? It's not too it's not heavily cumulative as like physics, for example. But still, like you gotta remember some of your stuff, guys. Okay? So the highlight of the stuff, guys, for today is the enzymatic reactions. Okay, so in order for the reactions to occur, even for this reaction, okay, like reaction like this, okay, it must have an enzyme. It's not gonna happen on its own in bio. Okay, in bio, every reaction must be catalyzed. Okay, which means what? Yeah, there must be an enzyme, okay, present for any reaction, okay? To what? Like, the reason is what? As a regulation perspective, okay? You don't want the reaction to happen every day, okay? You don't want these reactions to happen every day. You want to control it. So you want to run the process when it's needed, okay? Like, dude, like, it's one of the things, right? So, the perfect example, guys, okay? To kind of bring it up, right? So, I don't want to like waste too much time here, guys, but so in terms of you know, like in terms of guys like activation of this process, like okay, so enzyme must be present, okay, in order for the process to occur. So perfect example is what, guys? Have you heard of people, guys, who's lactose intolerant, guys? Have you heard of them, guys? Lactose intolerant means like you can't eat milk, right? So why is that the case, guys? Just so you know, because typically, guys, in your large intestine, there's what? Large intestine, there is a prokaryotic cells that live there who lives off of what, guys? Like waste, like the food that you don't eat, okay? Such as like cellulose, stuff like that, right? So now, if someone, okay, failed to break down lactose, okay, which is a lot in the milk, right? Then what happens? If you're failed to break down, it will not get absorbed by the small intestine and what goes into a large intestine. And large intestine is what, guys? They're gonna feast on those guys. Okay, so you understand, guys, what I'm saying? So they're gonna feast on them. So, which means, oh yeah, there's a food. Okay, they're gonna start to break it down and what? Start to run fermentation, which produce gas, and that's why they have a lot of gas build up in their body. Because why? Yeah, dude, there's a lot more nutrient for them compared to what it usually is for them, right? So what what I'm saying is, guys, okay, like activation process, okay? So these prokaryotic cells, this is something very, they talk about this a lot on the API, guys, something they call lac operons, okay? Where, the activation occurs, okay? Activation, for example, okay? So there's activation, so lac operon is a portion of the DNA, okay, for the prokaryotic cells where they activate this part of the DNA, okay, which will break down lactose into galactose and glucose, okay? So that when it's present, so this can be only activated, guys, when lactose is present, okay? And then the enzyme, which is galactosidase, break it into glucose and galactose. So what I'm trying to say here is, every process that occurs, okay, in your body or even a cell, they must be what? Regulated, okay? Without an enzyme, in this case, what? It's a beta, okay, galactosidase. Galactosidase. That's an enzyme that will break down lactose into glucose and galactose, okay? Without this enzyme, this will never happen, okay? They must be regulated, okay? So, so how do they do it? And okay, so why? Okay, you gotta understand, guys, how is the enzyme assist the reactions to happen that quickly, okay? What are they, So as we talked about, guys, these enzymes, well, like galactosidase, for example, guys, and keep in mind, this enzyme only does one thing. They got one job only, okay? They got one job, like you guys, right? What's your job, guys? Study, okay? You guys have to study and do all the tests, right? That's your job, okay? Right now, I mean, not only that's one thing that you do, but that's like the main job you guys have, right? So they got one job, okay? And they're pretty good at this, okay? They will see this and what? Turn that guy into these two, okay? So I mean, and without this enzyme, this will not occur, okay? So you gotta how? How does it happen? So keep in mind, guys, okay? If you got lactose here, okay, so let's assume, I don't think this is an exothermic process or exergonic process, but let's assume, okay? It's the process of lactose, okay, into like glucose and galactose. So that's the process, right? But this is what, guys, there is always what? Energy activation process. There's always energy activation. You gotta put enough energy to what? Overcome this process, right? Just so you know. So, but how can enzyme assist? Or you guys probably know, right? Oh yeah, if you have an enzyme, this goes down, okay? And then now the reaction can happen more easily. So, in a higher level, right? If you have an enzyme, what happens, guys? If you have an enzyme, what happens, guys? It lowers the energy of activation. I mean, if you're coming from chem, guys, I mean, this is something that you've probably seen a lot. Like, yeah, in chemistry, right? By adding a catalyst, how does it lower the energy of activation, guys? How? Yeah, what do you think?
Is it like it provides an alternate reaction pathway?
Exactly, it provides a different pathway for the reaction where it has a lower energy of activation, okay? So, but it's a little bit different, guys, okay? I mean, that's how it goes down, okay? But now, in chemistry, right, in AP Chem, which you guys did, catalyst can be anything. It could be an element. It could be like Cl. That's a catalyst, okay? But in bio, catalyst a lot of times is what? Is given as an enzyme, okay? Which is a protein, okay? So so how can these guys assist this process, okay? So you gotta understand. So So these enzymes are typically what? Globular proteins. Globular proteins, right? Which has what? Very specific shape. Okay, very specific shape. Okay, so I'm gonna give you guys a structure, guys. I mean, like, you know, I'm sorry, guys. Like, my drawing is always not the best, but this is the shape, guys. Okay, of the enzyme. Okay, globular protein, which is a galactosidase. Okay. So now what happens is, okay, this is now, guys, lactose. Lactose, guys, has two parts. So this is so there's this is glucose, and now this other part is galactose. Okay. So I'm sorry, guys. Okay, first, guys, the enzyme is much larger than the substrate. By the way, I'm just giving an example here. Okay. So this is a globular protein. Okay, and this is like, oh, what do we call this guy? Oh, whatever. We call this what? Active site. Okay, it's called the active site, right? And this is what, sometimes they call this what, like the lactose as what, substrate. Okay. This is a bioterm. Okay. Like, dude, in chem, guys, they don't say substrate. Okay, they call reactant, right? So when you look at this, guys, what happens, guys? So the fit of the active site to this is what? Like, yeah, very specific fit. So the shape of this is what? Like, very specific to what? Very specific to the shape of the substrate. Okay. Yeah, what do you got? Yeah, where you going? So if you look at it, what happens, guys? So yeah, this active site is what? Specific to the shape of the substrate. Okay. So which means what? If the substrate looks like this, they can't go in. Okay. That's a specificity that we're talking here. Okay. So you gotta understand, guys. Okay. So this guy will bind to the active site. So what you have to understand is this, okay? When this binds to the active site, right? What kind of binding are we talking? So there must be some kind of attraction, okay? Some kind of bond, maybe? What kind of bond is it? What kind of bond should be in the active site with the enzyme to the substrate, okay? AP, I'm not sure, but it's what? Typically what? Hydrogen bonding, like hydrophobic interactions, and what? Ionic. Okay, so these are typical bonds that will be made with the substrate, with the protein. Okay, like it's not covalent. Okay, covalent bond, no. So non-covalent, guys, hydrogen bonding, hydrophobic interaction, hydrophilic interactions, what? Ionic. These are the bonds that'll be what? Made with the substrate, with the protein. Okay, you gotta know AP, like, but keep in mind, guys. So how, like, okay, that's, like, you guys probably know, like, oh, it goes in, and what happens, guys? It what? Comes out separated now. Okay, that's what happens, right? But, like, do you get understand, like, how does that lower the energy of activation? Is that providing a different pathway for the whole process? It's kind of hard to say, okay? But now, how can an enzyme, okay, assist of lowering the energy of activation? Okay, like, that's the question you have to ask yourself. So, one thing to understand is what? So, first thing is, the enzyme providing the templates where the reaction can occur. Okay, so you provide them little space. So like, just like you guys come here, you know, like you guys meet with me and like I teach you guys, it provide a location where the reaction can potentially occur. Okay, so you know, so that's where it provides what? Little template area. Okay, where the reaction can be possibly happening. Okay, that's one thing to know. Okay, and second is what? Okay, and second is when they try to wiggle themselves into the active site, what happens, guys? It changes the conformation of substrate a little bit, okay? Which means what? Like when they go in, what happens, guys? It kind of twists and turns and stretch the substrate a little bit, okay? Which means what? Which potentially what? Lead to breaking of what? The two, okay? Turning into a product. So this is AB, turning to what, guys? A plus B, right? So as it goes in, what happens, guys? Like, dude, like goes in, like what happens? Like they're twisting and turning and stretching it out to what? So that what? It leads to what? Breaking of what? The two A and B into A plus B. Okay, do you understand what I'm saying? So like is what too? Is what, guys? It what? Stretching, okay, and potentially breaking, okay, the bond of the substrate or creating a bond. Okay, depends on the case, right? which can what? Which all this what? Lowers potentially what? Enzyme activation process. What else, guys? What else? Sometimes, guys, also as well, right? Active site. So for example, guys, okay, like okay, so some also what, guys? Active site can be what, guys? It can have a lot. So on here, on this portion of the active site, it can be what, guys? Very like acidic. It has a lot of acidic amino acids. Okay. What do you think, guys? On the active site is what? The pH is lower. Okay. So which means what? A third one is what, guys? It can provide what? Microenvironment. Okay, for specific reactions to occur. You know what I'm saying, guys? So yeah, dude, the reaction is what? Yeah, we might this breaking of the glucose and galactose, okay, potentially maybe work a little better if the environment is acidic, okay? And the active site, based on the side chains, what can provide the microenvironment can provide what? Low pH, okay? So yeah, it goes in. Oh, pH lower inside here. What happens, guys? A better chance of what? This to break, which is what potentially lowers what less energy needed to break them down. Okay, so yeah, that's also right. So that's the third one. What's the fourth one, guys? Just like other catalysts, right? The active site can potentially make covalent bond with these guys. Okay, and potentially what? Involved in a reaction with the substrates. Okay, so they actively involve. They actively what? Involved in a reaction. Okay, the active site, and it goes back to what it was before. Okay. But now, that's how the enzymes assist, okay, a reaction and lowers the energy of activation. Is what? Again, they provide the template for the reactions to occur, okay, and also what, guys? So if you look at it, guys, as the substrate goes in, it changes the conformation of substrate a little bit, which can potentially lower the energy of activation as well. And it also what, guys? It provides a microenvironment, such as what I mentioned, like it could be acidic or basic, which is favorable for the reactions to break down. And what? It also can what? Actively make covalent bond. That doesn't really happen. And also what? Assist in breaking down of the substrate. Okay, that's 4 things the enzymes will do to assist with the breaking down. Okay, which can potentially lower the energy of activation. Okay, so your teacher mentioned this guy. I don't know. You know, like it's good to know. So your teacher may, your school guys, they might just say, oh yeah, enzyme, guys, it lowers the energy of activation. Next chapter. It might not be good enough, guys, for higher end testing, by the way. Okay? You gotta know exactly what the hell is happening with the enzymes, okay? Which is not very similar to what you guys learn in AP Bio, AP Chem, right? AP Chem. So AP Chem is what? Oh yeah, just like what this guy said, it provides different pathway of the reaction. That might not be the case here, okay? So in bio is more what, guys? Activation and regulation and deactivation, okay? Any questions, guys? Let me know how the enzymes react with the substrate. Let me know. I know it's a mess here, but hope you guys understand what I'm saying. So, one thing to understand, guys, here now is the rate at which the enzymes react. Okay, this is something you probably heard, guys, but we'll discuss this a little more in detail for you guys for the AP Bio. Okay. Have you seen this curve, guys, by the way? Okay? Like, yeah, have you seen this, guys? I mean, dude, like, you don't have to be like super smart to like see this curve, right? This is what? The velocity, okay, or speed of the reaction, and this is what? Substrate level, okay? Which is the amount of reactant. Okay? This is called the Michaelis-Menten curve. Okay, curve. Okay, so Menten. It's in the reading, I think. Okay. This represents speed of the reaction. Okay, this represents what? Amount of substrate. Okay, like how much substrate you put in, how much reactant you put in. So now, this where they kind of level off, guys. So if you increase more, if you add more substrate, the rate goes up. Okay, and then what happens, guys? This becomes what we call the Vmax. Okay, which is a maximum speed. Okay, so keep in mind, guys. Right? There's a limitation of what? There's a limitation of enzymes. Okay, enzymes. I mean, you cannot just produce it like that easily. Okay, either let's assume this is what? This is assuming you have a limited amount of enzymes. Okay, so now you can understand the graph, right? This is what? Independent variables. Okay, this is dependent. Other variables are what? Locked down, not changing. Okay, the temperature stay the same. pH stay the same, and the number of enzymes stay the same, and this is what? The amount of substrate, okay? Only what goes up is what? Amount of substrates. Yeah, dude, I'm sure you've seen this, right? So up to a certain point, the rate goes up, okay? And after that, what happens, guys? It doesn't change, okay? And that's one, I mean, yeah, this is a guy, okay? I can do this. It's like I can get a little prize with this, okay? If I go back 50 years back, okay? If I go back in time, guys, I mean, I'm gonna bring all these equations and like, oh, 5 little prizes, okay? There's an equation for this, by the way. If I'm not mistaken, guys, the V is equal to Vmax plus the substrate amount times. Sorry. You guys don't have to do the equation. AP guys, you don't have to do the equation, guys. By the way, this is Km plus the substrate level. Okay, that's the equation for this, guys. And we'll discuss what Km is. So Km is what, guys? This amount of substrate to reach half of Vmax. Okay, that's what we call, guys, Km. Okay, so if you put in this much substrate, guys, that's what we're gonna reach, what, half of the maximum amount of speed, guys, can reach. Okay, that's the equation for the Michaelis-Menten equation. Okay, that's the equation, guys. Okay. So now, if you're one of those math people, okay, if S is a big number, okay, think about it. If S is a big number, okay, but what the hell happens, guys? We'll do that. Vmax over what? S over S. Okay, because Km becomes very negligible, guys. You know what happens, guys? Then it becomes what? V is to the Vmax, which is around there. Okay, so you know, yeah, like math-wise, if you're one of those math people, okay, if you don't know, it's fine. AP Bio, like they don't really teach you a lot of math stuff, okay. They don't, this equation you will never see, or the teacher will never bring it up. Okay, maybe like on those teachers' little spreadsheet or something, they might have it, but you'll never use it. So reason I'm bringing it up, guys, okay, is you have to understand. So we have few minutes, guys. You have to understand inhibitions. I have a little story, guys. I don't think we can talk about today. Inhibitions, okay. So there is what we call competitive inhibition, okay, and there's something called non-reversible inhibitions. Competitive inhibition, okay. There's also what we call non-competitive inhibitions. Okay, inhibition is what, guys? Like, dude, like you inhibit the reactions. You just want to stop the reaction in some way. Okay. So now, non-reversible reaction is what, guys? Yeah. So what the hell is a non-reversible reaction? You're more likely what? Either changing the enzyme, okay, to what? So that what? It will never act as how it should act. Like meaning like you're trying to what? Denature the protein, the enzyme. You know what I'm saying, guys? Here. So the enzyme is like shape is like this, okay? For example, right? You put in something in there as an inhibition. You want to stop it. Okay? You want to stop the reaction. Okay? You put in something that blocks. Okay? That covalently bonds. So the term is what? Covalent bonds. So I don't know why, guys. When you guys do bio, they assume covalent bond as a very strong bond. Okay? Like you gotta understand. Okay? Covalent bond is the strongest thing ever. You know? So the distinct covalent bonds that what incorporates the active site of what? It blocks substrate from going in. Okay, that is an example of what, guys? Non-reversible. Okay, inhibition. Okay, you want to stop that. Okay, yeah. How would this graph change? Yeah. What do you think, guys? How would this graph change? What do you think? So if there is a non-reversible inhibition which blocks the enzyme overall, what happens, guys? This would be what? Practically nothing. Okay, the reaction will not occur. You know what I'm saying? Or very, very small, like like this. You know what I'm saying, guys? Here. Yeah. What about competitive inhibition? What the hell is this? Competitive. So there is a substrate. Okay. So you want to control this, right? You want to control this process. So this is a substrate. Okay. This is a substrate, but you're putting in something. Okay. You want to stop. You want to lower the reaction, right? You're putting in something in there that what? Competitively fight for the active site. Okay, that's competitive inhibition, which means what? They're competing for the active site. Okay, so you gotta inhibit the process by what? Acting, putting in what, guys? Competition there. Okay. So now, if there's a competitive what? You got 2 minutes. Okay. Competitive inhibition. What do you think will happen, guys? What do you think? The Vmax. So I mean, again, if you add, like, dude, if you put in, for example, guys, okay. If you have like 100 of these guys, if you have 10,000 of these guys, what happens, guys? I mean, yeah, this will what? outweigh this guy, okay? Which means that overall, the Vmax will not be what? Impacted, okay? But which means what? This will be more like this, okay? The Vmax doesn't change, but it takes longer time to get there, okay? Because what? I do, like, if there's a million, if you influx like millions of guys, what happens, guys? Yeah, this means nothing, okay? It's not competitive anymore. You know what I'm saying, guys? Here? So, that's one thing to understand, guys, competitive inhibition, guys. And non-competitive, I mean, so I guess I gotta let you guys go. I have nothing class, guys, but it's a non-competitive. Like, yeah, non-competitive lowers the Vmax. We'll talk about it a little more next week. But again, guys, there's a homework assigned. Okay, so check your email, guys, with the homework. I'll try to send you guys an email, guys, hopefully by today, with the information. You guys have the logins, and I'll send you login for some of the folks here. And then I'll give you guys a link for the recordings here. Okay, all right, guys. I'll see you guys next week, guys.